Imagine, for a moment, that we are standing in a quiet, candlelit room in the late 19th century. A young mother is weeping over a cradle. Inside lies a newborn, her second child, but the infant is strangely pale, its skin swiftly turning a sickly, jaundiced yellow, its breathing shallow and labored. The doctors stand by, helpless, murmuring about bad blood or familial curses. They have no tools to stop the tragedy unfolding, because the enemy isn't a bacteria or a virus. The enemy is the mother's own body. For thousands of years, this scene played out in cottages and castles alike. It was a silent, heartbreaking mystery where a woman's blood turned into a weapon against her own offspring. This wasn't magic, and it wasn't a curse. It was a microscopic war fought on the surface of red blood cells, a war dictated by a tiny, elusive protein that was missing from some and present in others. This is the story of the rhesus factor, the Rh-negative blood type, a biological anomaly that has baffled scientists, fueled conspiracy theories, and ultimately saved millions of lives once its code was cracked. Over the course of this story, we are going to unravel the red mystery of this genetic trait. We will travel from the sterile laboratories of Vienna to the rugged mountains of the Basque country and back through the mists of human evolution to ask where this blood type came from. We will look at the science of why negative blood is so unique, explored through the lens of history's great medical detectives, and we will touch upon the strange modern cultural phenomenon that suggests Rh-negative people are somehow different. But before we can understand the negative, we must first understand the blood itself and the centuries of darkness that preceded the light of discovery. For most of human history, blood was viewed not as a biological fluid, but as a mystical essence. It was the life force, a divine substance that carried the soul. Ancient healers, from the Greeks to the medieval barber surgeons, believed that health was a balance of humors and blood was the most sanguine of them all. But they treated it as a singular universal substance. To them, blood was simply blood. It was red, it was warm, and it was vital. If a soldier bled out on a Roman battlefield, or a knight lost a limb in the Crusades, the idea of replacing that lost volume with someone else's blood was terrifying and strictly forbidden, largely because early attempts almost always ended in disaster. There are grim records from the 17th century of physicians attempting to transfuse blood from sheep or calves into humans, operating under the naive belief that the gentle nature of a lamb might cure a madman. The results were catastrophic. The patients would suffer agonizing fevers, their urine would turn black, and death usually followed within hours. The human body violently rejected the foreign invader. The medical community recoiled, banning transfusions for centuries. It seemed that the secrets of the blood were meant to remain locked away, a divine boundary that man was not supposed to cross. It wasn't until the turn of the 20th century that the first key to this lock was found. The year was 1900, and the setting was the imperial city of Vienna, a hub of art, philosophy, and rapidly advancing science. In a laboratory that likely smelled of formaldehyde and ozone, a reserved and meticulous Austrian biologist named Karl Landsteiner was conducting a simple experiment that would eventually win him the Nobel Prize. Landsteiner was bothered by the inconsistency of blood. He knew that sometimes, rarely, a transfusion between two humans worked, but most of the time, the recipient's blood would clot and clump, a reaction called agglutination. He decided to test the blood of his own laboratory staff. He took samples from 6 people, separated the red cells from the golden plasma, and then began mixing them in different combinations in glass petri dishes. As he peered at the mixtures, likely under the warm glow of a gas lamp, he noticed a distinct pattern emerging. In some dishes, the red liquid remained smooth and free-flowing. In others, the red cells clumped together like curdled milk. This clumping wasn't random chaos, it was a predictable chemical reaction. Landsteiner realized that on the surface of the red blood cells, there were microscopic markers, little chemical flags. If you mixed blood with the same flag, they got along. If you introduced a foreign flag, the plasma attacked it. He labeled these flags A and B. If you had the A marker, you were type A. If you had the B marker, you were type B. If you had neither, you were type C, which was later renamed type O for un, the German word for without. This was the dawn of the ABO blood group system, the foundation of modern hematology. It was a monumental breakthrough. Suddenly, doctors understood why those ancient transfusions had failed. You couldn't give type A blood to a type B person. Their immune system would recognize the A flag as an invader and send out antibodies to destroy it, causing the blood to clot and the patient to die. Landsteiner had drawn the first map of the human internal landscape. It seemed that the mystery was solved. The medical world rejoiced, believing they had finally conquered the danger of transfusion. Surgeries became bolder and lives were saved. However, as the decades rolled on, a quiet unease began to settle over the hematology wards of Europe and America. The ABO map was excellent, but it was incomplete. There were ghosts in the system. Occasionally, doctors would follow Landsteiner's rules perfectly, transfusing type O blood into a type O patient or A into A. And yet, disaster would still strike. The patient would react violently, spiking a fever, shaking with chills, their body rejecting the seemingly compatible blood. It was a statistical anomaly, occurring often enough to be terrifying, but rarely enough to be dismissed as surgical error or contamination. This was particularly evident in obstetrics, the branch of medicine dealing with childbirth. There was a specific, recurring tragedy that haunted maternity wards. A woman would have a healthy first child, but her second or third pregnancy would end in heartbreak. The baby would be born swollen, pale, or yellow with jaundice, a condition then known as erythroblastosis fetalis. The infant's blood was being destroyed from the inside out. Even more baffling, if that mother needed a blood transfusion after a difficult birth and the doctors gave her blood from her own husband, who matched her ABO type perfectly, she might suffer a severe, life-threatening reaction. It was as if the woman's body had been sensitized to something, primed to attack. But what? If the A and B antigens matched, what else was there? The map Landsteiner had drawn in 1900 had huge blank spaces that no one could see. The blood was hiding another secret, a deeper layer of identity that was invisible to the tests of the time. By the late 1930s, the pressure to solve this riddle was mounting. War was looming on the horizon in Europe, and military leaders knew that safe blood transfusions would be the difference between life and death for thousands of soldiers. The anomalies could no longer be ignored. The scientific community needed to find the X factor, the hidden variable that was causing compatible blood to turn deadly. This leads us to the concept of the antigen, generally. To understand the gravity of what was about to be discovered, we have to visualize the red blood cell not just as a carrier of oxygen, but as a heavily armored vessel patrolling the bloodstream. The surface of a single red blood cell is like a complex terrain, studded with proteins and sugars. The A and B antigens are like large, obvious banners flying from the ship's mast. But the surface is also covered in hundreds of other, smaller structures. Most of these are universal. Almost every human has them, so our immune systems ignore them. They are recognized as self. The mystery lay in finding a structure that wasn't universal, something that some people had and others lacked. It is the nature of the human immune system to be a ruthless xenophobe. It tolerates absolutely no strangers. Its primary job is to distinguish self from non-self. When it encounters a protein it doesn't have in its own inventory, it doesn't ask questions, it launches a nuclear strike. The tragedy of the blue babies and the failed transfusions suggested that there was another banner flying on some blood cells, a banner that Landsteiner's original experiment hadn't detected. For the mother whose baby died, the tragedy was a result of this invisible war. Her body, lacking this mysterious protein, had encountered it in her baby, inherited from the father. During the first pregnancy, her system merely noticed the intruder. But, like a sleeping dragon, her immune system woke up. It forged weapons, antibodies, specifically designed to target that protein. When the second pregnancy occurred, the dragon was waiting. Her antibodies crossed the placenta, the very organ designed to nourish life, and attacked the fetus's blood as if it were a virus. This was the state of affairs as the world entered the 1940s. The medical mystery was a locked room. We knew the what, hemolytic disease, transfusion reactions, but we did not know the why. We needed a new key, And surprisingly, that key would not come from humans at all, but from a small brownish-gray primate native to Asia, the rhesus macaque. The answer was hiding in our evolutionary cousins, waiting for Landsteiner, now older and working in New York, to return to his microscope and look deeper into the red mystery than anyone had ever looked before. The stage was set for the discovery of the rhesus factor, the defining divide between the positive and the negative, a distinction that would explain why some blood is a universal healer and some a potential hazard. It was 1940, and the world was already consumed by the fires of the Second World War. While nations were battling for territory across Europe, a quieter but equally significant battle for knowledge was taking place in New York City at the Rockefeller Institute for Medical Research. Carl Landsteiner, the man who had mapped the ABO blood groups 40 years earlier, was now in his 70s. He had retired from his post, but not from his curiosity. He was joined by a brilliant younger colleague, Alexander Wiener. Together, they were hunting for that invisible variable, the ghost in the blood that was causing unexplained transfusion reactions. Their subjects for this historic experiment were not human patients, but rhesus macaques. These small, resilient monkeys were common in research laboratories because their physiology was remarkably close to humans. Landsteiner and Wiener had a theory. Perhaps there was an antigen in human blood that was similar to one found in monkeys. If they could identify it in the primate, they might be able to find its echo in man. The experiment was elegant in its simplicity. They took red blood cells from the rhesus monkeys and injected them into rabbits and guinea pigs. The immune systems of these small animals reacted exactly as expected. They recognized the monkey blood as a foreign invader and produced antibodies to destroy it. Landsteiner and Wiener then extracted the serum, the antibody-rich part of the blood, from the rabbits. They now had a chemical weapon specifically designed to target and clump rhesus monkey blood cells. They called this anti-rhesus serum. Now came the crucial step. They took this anti-rhesus serum, which they knew attacked monkey blood, and mixed it with human blood samples from New York donors. They held their breath, waiting to see if the human blood would react to the monkey-targeting antibodies. The results were startling. In about 85% of the human samples, the blood clumped together. The rabbit antibodies designed to fight monkey blood were attacking the human cells. This meant that 85% of the humans had the same protein on their red blood cells as the rhesus monkeys. Landsteiner and Wiener named this protein the rhesus factor, or Rh factor for short. Those who had it were labeled Rh positive. But what about the other 15%? In those samples, the blood remained smooth and liquid. The antibodies ignored them. These people did not have the monkey protein. Their blood cells were clean of this specific marker. They were labeled Rh negative. This was the eureka moment. The ghost had been found. The medical community suddenly had a new map. A person wasn't just type A or type O anymore. They were A positive or A negative, O positive or O negative. The addition of this simple plus or minus sign explained the decades of tragic accidents. Landsteiner and Wiener realized that the Rh negative individuals were the vulnerable ones in a transfusion scenario. If an R negative person, someone without the protein, received blood from an R positive donor, someone with the protein, their immune system would react just like the rabbits did. It would see the Rh protein as a dangerous foreign invader and launch a massive attack. The first time this happened, the reaction might be mild, a sensitization event. But the immune system has a long memory. If that person received Rh positive blood a second time, the antibodies would swarm the donor cells, rupturing them instantly, clogging the kidneys, and sending the patient into shock. This discovery immediately revolutionized battlefield medicine. As the war raged on, doctors began testing soldiers not just for ABO, but for Rhe status. They realized that giving universal O-positive blood to an O-negative soldier was a roll of the dice that could kill him. The definition of the universal donor shrank. Only O-negative blood, blood lacking both the AB antigens and the Rh protein, could truly be given to anyone safely. O-negative became the liquid gold of emergency medicine, the only safe bet when there was no time to type a patient. But the implications went far beyond the battlefield. The discovery of the Rh factor solved the heartbreaking mystery of the blue babies and the mothers who lost child after child. It was a revelation that linked the laboratory directly to the womb. It turned out that the lethal womb scenario was a simple case of genetic incompatibility. If an Rh-negative woman mated with an Rh-positive man, the laws of genetics dictated that the baby had a high chance of being Rh-positive, inheriting the father's protein. This meant the baby carried a substance in its blood that the mother's body considered alien. During a normal pregnancy, the mother's and baby's blood supplies are kept separate by the placental barrier. But during childbirth, or if there is trauma, a small amount of fetal blood often leaks into the mother's bloodstream. For an Rh-negative mother, this leak is the trigger. Her immune system spots the Rh-positive cells from the baby and flags them as rhesus virus. She begins to produce anti-Rh antibodies. The first baby is usually born safely before this immune response fully kicks in. However, the mother is now sensitized. She carries the weapon in her blood, waiting for a target. If she gets pregnant again with another Rh-positive baby, her antibodies are ready. They are small enough to cross the placenta. They hunt down the fetus's red blood cells and destroy them. The baby becomes anemic. Its heart struggles to pump the thinning blood, and its liver swells as it tries frantically to produce more cells. This was the cause of erythroblastosis fetalis, now known as hemolytic disease of the newborn, HDN. Landsteiner and Weiner's discovery in 1940 didn't essentially cure the disease overnight, but it illuminated the mechanism. It allowed doctors to predict which pregnancies were at risk. If a mother was Rh-negative and the father was Rh-positive, they knew to be vigilant. It was the first step toward a treatment, though that treatment was still decades away. The connection to the rhesus macaque was not just a medical footnote, it was a profound biological statement. It highlighted a shared evolutionary heritage. The fact that 85% of humans shared this protein with monkeys suggested a common ancestor deep in the primate lineage. We were, biochemically speaking, part of the same family tree. But the 15%, the Rh-negatives, posed a new question. Why did they lose this protein? Or, conversely, did they never have it? In the grand scheme of evolution, losing a protein usually happens for a reason. Was it a random mutation, a beneficial adaptation, or something else entirely? The existence of Rh-negative blood is a puzzle because, in a purely Darwinian sense, it looks like a disadvantage. Before modern medicine, Rh-negative women had a significantly harder time passing on their genes because of the high rate of infant mortality in second and third pregnancies. A trait that kills your offspring should, theoretically, be weeded out by natural selection. Yet, Rh-negative blood persisted. It survived in the human population for thousands of years. This persistence suggests that being Rh-negative must offer some hidden advantage, some counterbalance to the reproductive risk. Perhaps it offered protection against a disease that ravaged the ancient world. Perhaps it was tied to other traits that gave carriers a survival edge. Landsteiner's monkeys had revealed the what, but they had left us with a massive why. The scientific community in the 1940s was scrambled. They had to update every medical textbook, retrain every nurse and doctor, and overhaul the entire blood banking system. The terms Rh-positive and Rh-negative entered the global lexicon. But amidst the flurry of medical reorganization, anthropologists and geneticists began to notice something strange about the distribution of this trait. It wasn't evenly spread across the globe. In Asian and African populations, the Rh-negative blood type was extremely rare, found in less than 1% of the people. Almost everyone was Rh-positive, sharing the protein with the macaque. But in Europe, the numbers were different. In some regions, 15%, 20%, or even 30% of the population was Rh-negative. This geographic anomaly hinted that the Rh-negative mutation was not a global human event, but something that happened in a specific place, at a specific time, to a specific group of people. It was a genetic breadcrumb trail leading back into the mists of European prehistory. As researchers began to map the frequency of the gene, the trail pointed them toward the secluded valleys of the Pyrenees Mountains, to a distinct and ancient people known as the Basques. The discovery of the Rh factor was more than just a medical triumph. It was the opening of a door into the deep history of human migration. It turned blood into a historical document. Landsteiner and Wiener had set out to make transfusions safe, but they ended up handing us a map to our own diversity. They showed us that beneath the skin, we are divided by an invisible chemistry, a legacy of ancient mutations that still dictates life and death in the delivery room today. The monkey blood experiment had changed everything, proving that sometimes, to understand what makes us human, we have to look at what separates us from the rest of the animal kingdom. To fully grasp the horror of what was happening before we understood blood types, we have to look away from the microscope and into the nursery. We must understand that for an Rh-negative woman carrying an Rh-positive child, the womb, universally revered as the ultimate sanctuary of life and protection, transformed into a biological trap. This condition, which Landsteiner and Wiener's discovery finally illuminated, was known medically as erythroblastosis fetalis, but to the families who endured it, it was simply a curse that stole their children one by one. It turned the miracle of birth into a recurring nightmare of silence. The mechanism of this tragedy is a masterclass in the ruthless efficiency of the human immune system. As we touched upon, the immune system is designed to seek and destroy. It does not understand context, it only understands codes. When a mother is Rh negative, her blood lacks the D antigen. Her immune system views this D antigen not as a harmless protein, but as a threat on the level of smallpox or the plague. During a first pregnancy with an Rh-positive baby, the two bloodstreams usually remain distinct. The baby is safe inside the amniotic sac, separated by the placental barrier. The mother is like a fortress that doesn't yet know the enemy is camping just outside the walls. However, the moment of birth is violent and messy. As the placenta separates from the uterine wall, there is almost always a mixing of fluids. A few drops of the baby's cord blood enter the mother's circulation. It is a tiny amount, perhaps no more than a teaspoon, but it is enough. The mother's white blood cells intercept these foreign Rh-positive cells. They analyze the surface proteins, identify the intruder, and sound the alarm. The mother's body begins to manufacture antibodies, specifically immunoglobulin G or IgG. Here is the crucial lethal detail. Unlike other antibodies, such as those for ABO blood types, which are large and clunky, the Rh antibodies are tiny. They are sleek, efficient hunters. And because they are so small, they possess a terrifying ability. They can cross the placenta. The mother is now sensitized. She is a walking weapon system, armed and waiting. When she becomes pregnant a second time with another Rh-positive child, the tragedy begins silently, months before the due date. The mother's IgG antibodies drift across the placental barrier, entering the fetus's bloodstream. They are looking for the D antigen. When they find it on the developing baby's red blood cells, they attach themselves to the cell surface. This marks the cell for destruction. The baby's own spleen, doing its job of filtering the blood, recognizes these antibody-coated cells as damaged and destroys them. The fetus is now fighting a war on two fronts. First, it is losing blood cells faster than it can make them. This causes severe anemia. To compensate, the baby's bone marrow goes into overdrive. When that isn't enough, the liver and the spleen swell to massive sizes, trying desperately to churn out new red blood cells to keep the heart beating. This is why these babies were often born with distended, swollen bellies. Second, the destruction of red blood cells releases a byproduct called bilirubin. In a healthy adult, the liver processes bilirubin and excretes it. But the fetus's liver is immature, and it is already overwhelmed by the task of making new blood. The bilirubin builds up in the baby's tissues. This is a neurotoxin. It turns the skin and the whites of the eyes a deep saffron yellow, a condition called severe jaundice. If the levels get too high, the bilirubin crosses the blood-brain barrier and settles in the deep gray matter of the brain, causing a specific type of permanent brain damage known as kernicterus. The presentation of these infants at birth was heartbreakingly distinct. Some were born with hydrops fetalis, a condition where the anemia is so severe that the baby's heart begins to fail in the womb. The baby's body swells with fluid, accumulating in the lungs, the abdomen, and under the skin. They would be born still, pale, and incredibly swollen, as if they had drowned. These were the most severe cases, the ones where the war was lost before the baby even took a breath. Others were born alive but quickly deteriorated. This is where the term blue baby is sometimes conflated, though medically distinct. While blue baby syndrome typically refers to heart defects causing a lack of oxygen, cyanosis, the Rh baby was often a mix of pallor from anemia and the creeping yellow of jaundice. However, as their respiratory systems failed due to the fluid pressure and heart failure, they would indeed suffocate, turning a dusky blue-gray in their final moments. For centuries, this pattern baffled midwives and doctors. They saw a healthy first child, followed by a second child who died of jaundice, followed by a third who was born stillborn, followed by a series of miscarriages. It looked like a genetic decay, a weakening of the bloodline. Husbands blamed wives, wives blamed themselves. In royal families, where securing an heir was a matter of national security, this condition could topple dynasties. If a queen was Rhe negative and the king was Rhe positive, their reproductive future was a game of Russian roulette. Before the 1960s, there was no shot, no prevention. The only thing doctors could do was watch the antibody titers, levels, in the mother's blood rise, knowing the attack was happening. In the 1940s and 50s, after the Rh factor was identified, a heroic but gruesome treatment was developed, the exchange transfusion. If a baby was born alive but suffering from Rh disease, doctors would immediately insert a catheter into the umbilical vein. They would painstakingly remove the baby's poisoned blood, replacing it with fresh Rh negative blood from a donor. The Rhe negative donor blood was the key, since it lacked the D antigen. The mother's antibodies, which were still circulating in the baby's system, wouldn't attack it. This didn't cure the disease, but it bought time. It washed out the bilirubin and the antibodies, keeping the baby alive long enough for its own liver to mature and for the mother's antibodies to naturally flush out of its system over a few weeks. It was a high-stakes procedure. Doctors were essentially draining and refilling a newborn infant like a car engine. It carried risks of heart failure, infection, and air embolisms, but it was the only hope they had. Thousands of exchange babies grew up in the mid-20th century, walking miracles who survived only because someone else's Rh negative blood was pumped into their veins in the first hours of their lives. But for those who didn't make it to the exchange table, the outcome was grim. The lethal womb cast a long shadow over the demographics of the past. It acted as a natural check on the population of Rh-negative individuals. Mathematically, the gene should have been selected against. Every time an Rh-negative woman lost a baby to HDN, the Rh-negative gene, carried by the mother, and the Rh-positive gene, carried by the fetus, were both removed from the gene pool. However, because the Rh-positive gene is dominant and much more common, the loss hit the Rh-negative population harder in terms of reproductive success. This brings us to a compelling statistical mystery. If the Rh-negative blood type is a reproductive disadvantage, if it actively kills offspring in mixed marriages, why hasn't it gone extinct? Nature is brutal. It usually deletes traits that lower fertility. Yet, Rh-negative blood has persisted for tens of thousands of years. It suggests that the lethal womb is only half the story. There must be a counterweight, a reason why nature decided to keep this mutation in the rotation despite the high cost of infant mortality. To find the answer, we have to look at where this blood type is most concentrated. We have to leave the modern hospital and travel back to a specific geographic pocket of Europe. The distribution of Rh-negative blood is not random. It is clotted in specific populations, like a genetic footprint left by an ancient tribe. While the Rh factor was discovered in New York labs using Asian monkeys, the epicenter of the Rh negative people is nowhere near Asia. It is in the jagged, mist-covered peaks between Spain and France. The story of the lethal womb leads us directly to the Pyrenees paradox. Here, in one of the oldest and most isolated cultures in Europe, we find a population where nearly a third of the people walk around with Rh-negative blood, the highest concentration on Earth. If the womb was so lethal, how did this tribe survive? And more importantly, is this where the mutation began? The Basques are the guardians of the Rh secret, and their history may hold the key to the origin of the blood that kills its own. To find the epicenter of this biological anomaly, we have to turn our attention away from the sterile hospitals of the 20th century and zoom in on a map of the Old World. If we trace the distribution of Rh-negative blood across the globe, we see a distinct pattern. It is virtually non-existent in indigenous populations of the Americas, Australia, and East Asia. In Africa, it is rare. But as we move into Europe, the numbers begin to tick upward. The frequency hovers around 15% or 16% for most Caucasians. But there is a hot zone, a specific bullseye where the concentration of this blood type skyrockets to nearly double the European average. That bullseye is located on the rugged, mist-shrouded border between France and Spain in the ancient peaks of the Pyrenees Mountains. This is the home of the Basque people, or as they call themselves, the Euskaldunak. And they are the living, breathing heart of the Rh mystery. The Basques are an anthropological puzzle that has frustrated historians for centuries. They inhabit a region known as Euskal Herria, a lush, mountainous land that has historically acted as a fortress against invaders. While the rest of Europe was being overrun and remixed by waves of Romans, Visigoths, Moors, and Franks, the Basques remained stubbornly distinct. They preserved their culture, their laws, and most famously, their language. Euskara, the Basque tongue, is a language isolate. It is not related to French, Spanish, German, or indeed any other Indo-European language. It is a linguistic orphan, a relic from a time before the Tower of Babel, suggesting that the Basques were settled in these mountains long before the ancestors of modern Europeans arrived. But it is their blood that truly sets them apart. The Basque people have the highest frequency of Rh-negative blood in the world. Approximately 30% to 35% of the population tests as Rh-negative, specifically they have the phenotype DD, and up to 60% carry the recessive gene. Compare that to the global average, and it is a statistical mountain as steep as the Pyrenees themselves. Furthermore, they have almost zero occurrence of type B blood. In genetic terms, the Basques are as close as we can get to a pure control group for the European continent. This brings us to the Pyrenees paradox. How did a population with such a high rate of Rh-negative blood survive the brutal mathematics of the lethal womb? As we discussed, an Rh-negative woman faces high infant mortality if she mates with an Rh-positive man. In a mixed population, this usually drives the Rh-negative gene toward extinction. So, logic dictates that the Basques should have bred this trait out millennia ago. The answer to the paradox lies in their isolation. The curse of the Rh disease only happens when positive and negative mix. If an Rh-negative woman marries an Rh-negative man, there is no incompatibility. The baby is Rh-negative, the mother is Rh-negative, and the immune system stays asleep. Because the concentration of the gene was so high in the Basque country, and because they were culturally and geographically isolated for thousands of years, Rh-negative people were mostly marrying other Rh-negative people. The high frequency of the gene actually became its own protective shield. It created a genetic safe zone where the mutation could thrive without the reproductive penalty found in the outside world. But why was the concentration so high to begin with? This forces us to look at the timeline of human habitation in Europe. To understand the Basques, we have to go back to the last glacial maximum, the peak of the Ice Age, about 20,000 years ago. Imagine Europe covered in a thick sheet of glacial ice. The north, England, Germany, Scandinavia, was uninhabitable, a frozen wasteland. Human populations were forced to retreat south, seeking warmth. Anthropologists believe that the Franco-Cantabrian region, encompassing the Pyrenees and southern France, became a massive refuge. It was a bottleneck. Small bands of hunter-gatherers were squeezed together in this habitable pocket for thousands of years. It is highly probable that within this small, trapped population, the Rh-negative mutation occurred or was already present. Due to a phenomenon known as genetic drift, where traits in a small population can become dominant simply by chance, the Rh-negative blood type became standard for these people. They were the original Europeans, the Paleolithic hunter-gatherers who painted the caves at Lascaux and Altamira. When the ice finally melted about 10,000 years ago, these people didn't disappear. They expanded. They moved north to repopulate a thawing Europe, carrying their Rh-negative blood with them. However, a few thousand years later, a new wave of people arrived, the Neolithic farmers from the east, Anatolia and the Middle East. These farmers brought agriculture, domesticated animals, Indo-European languages, and Rh-positive blood. The history of Europe is essentially the history of these two groups colliding. The farmers slowly absorbed the hunters, diluting the original bloodlines. The Rh-negative trait became a minority, a recessive echo of the past. But in the Pyrenees, the mixing never fully happened. The Basques held the line. They stayed in their mountains, rejecting the farmers' language and their genes. They are, in essence, a window into the genetic makeup of the Cro-Magnon man. This connection to Cro-Magnon is not just romantic speculation. It is supported by physical anthropology. Early studies of Basque physiology noted distinct differences compared to their neighbors. They tended to have different cranial structures, specifically a long, straight nose and strong chin, features often associated with the Cro-Magnon skulls found in fossil records. While modern mixing has blurred these lines, the blood remains the most stubborn witness. The Pyrenees paradox also raises a fascinating question about the nature of the Rh mutation itself. If the Basques are the descendants of the original Europeans and they are heavily Rh negative, does that mean Rh negative is the original European blood type? Was the old Europe predominantly negative, only to be overwhelmed by a positive wave from the east? This flips the script on how we usually view the trait. We often call Rh negative a mutation or a deletion, implying it is a breakage of the normal code. But in the context of the Basques, Rhe positive is the intruder. To a Basque nationalist in the 19th century, the Rh negative blood was a badge of honor, a biological proof of their ancient purity. It was a sign that they were the Europeans, the ones who had been there since the beginning of time. Of course, this distinctiveness has historically been a double-edged sword. In the era of eugenics and racial science, the unique blood of the Basques attracted unwanted attention. During World War II, Nazi anthropologists were obsessed with the Basques. They sent teams into the mountains to measure skulls and take blood samples, trying to fit the Basques into their twisted Aryan theories. They were confused by a people who looked Aryan but spoke a non-Aryan language and had blood that didn't match the rest of the master race theories. The Basques, true to their history, offered little cooperation, remaining an enigma that refused to be categorized. The Pyrenees remain the spiritual home of Rh negative blood. It is the reservoir. If you are Rh negative, regardless of where you live today, be it Ohio, Melbourne, or Cape Town, it is highly likely that if you trace your DNA back far enough, you will find a thread leading to these mountains. You share a biological link with the people who withstood the Romans and the Moors. You carry the genetic signature of the Ice Age survivors. However, the Basques are not the only piece of the puzzle. While they hold the highest concentration, the Rh negative gene didn't just stay in the mountains. It moved. It traveled across the seas and deserts, leaving breadcrumbs in places that seem completely unconnected to Europe. To understand the full scope of this story, we have to leave the Pyrenees and track the migration of this gene into the wider world. We have to look at how this old European blood found its way into the veins of the Berbers of North Africa, the Black Jews of Ethiopia, and the royal houses of history. The paradox of the Pyrenees proves that Rh-negative blood is tenacious. It survives against the odds, but its journey was just beginning. As the ice melted and the tribes scattered, the gene for the negative blood began a great migration, hiding in the genotype of conquerors and traitors, silently passing down through generations, waiting to resurface and baffle the scientists of the future. If the Pyrenees Mountains were the reservoir, then the rivers of time broke the dam and sent this genetic trait flowing outward across the continents. The Rh-negative gene did not remain locked in the high valleys of Basque country. It hitched a ride on the migrations of restless tribes, painting a chemical trail that allows us to reconstruct the movements of ancient peoples long before the invention of the written word. By tracing the frequency of this blood type, we can see the ghost of a lost map, one that predates the borders of modern nations and hints at a time when the Atlantic facade of Europe and the coasts of North Africa were part of a connected maritime world. When we track the Rh-negative gene moving north from the Pyrenees, we run straight into the history of the Celts. It is no coincidence that after the Basques, the populations with the highest percentages of Rh-negative blood are found in the Celtic fringes of Europe, Ireland, Scotland, Wales, and Brittany. In parts of Ireland and Scotland, the frequency of the Rh-negative phenotype hits nearly 30%, rivaling the Basque numbers. This biological link suggests a profound kinship between the pre-Indo-European hunter-gatherers of the mountains and the ancestors of the insular Celts. Anthropologists and geneticists now largely agree that the Celtic people were not just a single wave of iron-wielding invaders from the east, as Victorian historians once thought. Instead, the genetic bedrock of the British Isles is ancient. The people of Ireland and Western Britain share a massive amount of DNA with the Basques. They are, in a sense, cousins separated by the Bay of Biscay. The Rh-negative gene in a modern Dubliner or a Highlander is likely a direct inheritance from those same Ice Age refugees who expanded northward as the glaciers retreated. They walked across the land bridge of Doggerland before it sank beneath the North Sea, carrying the D-negative allele into the mist-covered hills of the north. But the gene also traveled south, and here the story becomes even more intriguing. If you cross the Strait of Gibraltar into North Africa, into the rugged terrain of the Atlas Mountains, you find the Amazigh people, known to the west as the Berbers. The Amazigh are the indigenous inhabitants of North Africa, predating the Arab conquests by millennia. And like the Basques and the Celts, they possess a curiously high frequency of Rh-negative blood relative to their neighbors in the rest of Africa and the Middle East. This genetic anomaly has fueled decades of anthropological debate. How did a European blood trait end up concentrated in the mountains of Morocco and Algeria? The answer points to a prehistoric era where the Mediterranean was not a barrier, but a bridge. During the Paleolithic and Neolithic eras, there was significant gene flow back and forth across the strait. The ancestors of the Berbers and the ancestors of the Iberians were trading partners and perhaps even kin. The Rh-negative gene flowed south, embedding itself in the Amazigh population, surviving the isolation of the Atlas Mountains just as it survived in the Pyrenees. Even more mysterious is the case of the Guanches. These were the aboriginal inhabitants of the Canary Islands, living in complete isolation off the coast of Africa until they were discovered and largely wiped out by the Spanish in the 15th century. When early anthropologists studied the mummified remains of the Guanches and the blood of their few surviving descendants, they found a startling prevalence of type O blood and Rh-negative markers. The Guanches were tall, often fair-haired or blue-eyed, and culturally distinct from mainland Africans. Their blood linked them directly back to the Berber tribes, and, by extension, to the ancient European megapopulation. They were a time capsule, a pocket of Rh-negative carriers stranded on volcanic islands, preserving a genetic snapshot of the Stone Age until the Age of Exploration destroyed them. Tracing the gene further east, the signal begins to fade, but it doesn't disappear completely. It leaves breadcrumbs in the Levant and the Horn of Africa. There is a fascinating anomaly among the Beta Israel, the Ethiopian Jews. This community, which lived in isolation in the highlands of Ethiopia for centuries, has historically shown frequencies of Rh-negative blood that are higher than the indigenous African populations around them. This biological fact has been used to support their oral traditions of ancient migration from the Middle East, suggesting a genetic connection to the Semitic peoples of the north, who, in turn, had admixed with the populations carrying the Rh-negative trait. It is a testament to how blood can validate oral history. The gene acted as a silent witness to a migration that Scripture recorded, but history had often questioned. However, as we move into the heart of Asia, the Rh-negative signal drops into the void. In China, Japan, and Indonesia, the Rh-negative frequency is often less than 1%, sometimes as low as 0.3%. This creates a massive genetic gradient, a slope that runs from the high peaks of the Pyrenees down to the plains of the Orient. This gradient is one of the most important maps in human genetics. It essentially shows the expansion of the Neolithic farmers. About 9,000 years ago, farming was invented in the Fertile Crescent. These farmers began to migrate westward into Europe. They were predominantly Rh positive. As they moved, they interbred with the native hunter-gatherers, who were high in Rh negative. The further west the farmers went, the more their gene pool was diluted by the natives. By the time they hit the Atlantic Wall, the Basques and the Irish, the wave of farmers had lost its momentum. This is why the Rh-negative gene is highest in the west and lowest in the east. It is a map of resistance. The high frequency of Rh-negative blood in Western Europe is effectively a measurement of how much hunter-gatherer DNA survived the onslaught of the farmers. Modern genetics has given us a tool to overlay on this blood map, haplogroups. Specifically, the Y-DNA haplogroup R1b. This is the most common paternal lineage in Western Europe, and its distribution matches the Rh-negative map almost perfectly. Where you find high concentrations of R1b, in the Basques, the Welsh, the Irish, the Scots, you find high concentrations of Rh-negative blood. It implies that these traits traveled together, packaged in the bodies of a specific group of men who dominated the landscape of Bronze Age Europe. This migration story changes our understanding of what it means to be a carrier. Being Rh-negative is not just a random medical label. It is an inheritance from a specific ancestral ghost. It connects a person to the mammoth hunters of the Ice Age, to the megalith builders of Stonehenge, and to the fiercely independent tribes of the Atlas Mountains. It is the blood of the outliers, the people who lived on the edge of the known world. But this movement wasn't just about geography. It was about survival. As these tribes moved, they encountered new diseases, new climates, and new diets. The persistence of the Rh-negative gene during these arduous migrations suggests, again, that it wasn't just neutral cargo. It must have played a role. Did it help these ancient wanderers survive the cold of the Ice Age? Did it offer some immunity to the pathogens found in the dense forests of prehistoric Europe? The mapping of the gene also reveals the tragic dead ends. We look at the Guanches and see a population that was 100% isolated, where the gene flourished only to be extinguished by conquest. We look at the royal families of Europe, who we will discuss next, where the gene was concentrated not by migration, but by the artificial walls of class and incest, turning the blood into a curse. Tracing the gene is like following a faint red thread through the tapestry of human history. It ignores the borders drawn by kings and politicians. It tells us that the boundaries between European and African are more fluid than we think, bridged by the ancient movements of the Berbers and the sailors of the Mediterranean. It tells us that the Celts are older than the Celtic language. As we stand over the map of the world and stick pinheads into the high-frequency zones, the Basque Country, Ireland, the Rif Mountains of Morocco, the remote valleys of Switzerland, we see a pattern of retreat. The Rh-negative people were often the ones pushed to the edges, to the mountains and the islands, by the waves of newer, Rh-positive civilizations. They are the survivors of an older world, carrying a genetic code that has refused to be overwritten by 10,000 years of history. And nowhere was this refusal more dramatic or more consequential than in the marble halls of the palaces where the blue bloods tried to keep their bloodlines pure, only to find that their own biology was conspiring against them. When we hear the term blue blood, we immediately think of aristocracy. We picture the crowned heads of Europe, the dukes and duchesses, the elite lineage that separates the rulers from the commoners. Today, we use it as a metaphor for privilege, but the origin of this phrase is surprisingly literal, and it leads us straight back to the mystery of the rhesus factor. The term sangre azul was first coined by the nobility of Castile in Spain, the very same geographic region that borders the Basque country, the epicenter of the Rh-negative gene. In the Middle Ages, the Spanish nobility was obsessed with purity of lineage. They claimed descent from the Visigoths, the Germanic tribe that had swept down from the north after the fall of Rome. To prove that they had not mixed their blood with the darker-skinned Moors or other groups occupying the Iberian Peninsula, these aristocrats would roll up their sleeves and display the underside of their forearms. Their skin was so pale and translucent that their veins appeared vividly blue against the white flesh. This visible network of blue veins became a badge of honor, a biological passport, proving they were of the old stock, distinct from the rest of the population. Modern historians and geneticists have looked at this sangre azul phenomenon with fresh eyes. Given what we know about the high concentration of Rh-negative blood in the Basque and ancient Gothic populations, it is highly probable that this blue blood aristocracy was a reservoir for the Rh-negative gene. They were inadvertently signaling their genetic status, but while they wore their pale skin and blue veins as a symbol of divine right, their obsession with keeping their bloodlines pure would eventually unleash a biological curse that toppled dynasties and redrew the map of Europe. The history of European royalty is, in many ways, a history of reproductive failure. We often read about kings obsessed with securing a male heir, divorcing wives, beheading queens, and breaking from churches in a desperate bid to keep their name alive. We tend to attribute these struggles to bad luck, poor hygiene, or syphilis, but when we apply the lens of modern hematology to the royal family trees, a different, more specific pattern emerges. It is the pattern of blood incompatibility, the silent war between a mother's immune system and her unborn child. The royal curse often followed a terrifyingly specific script, one that we now recognize as the hallmark of rhesus disease or similar alloimmune conditions. The king and queen would marry. The first pregnancy would result in a healthy, vibrant child. The court would rejoice. The succession seemed secure. But then, the darkness would descend. The second pregnancy would end in a miscarriage. The third, a stillbirth. The fourth, a child born alive but sickly, yellowed with jaundice, dying within days. The fifth, a bloated, waterlogged stillbirth. This heartbreaking sequence, one healthy heir followed by a string of disasters, is the exact clinical progression of an Rh-negative mother becoming sensitized by her first Rh-positive baby. The first child survives because the antibodies haven't formed yet. But that first birth wakes the dragon. Every subsequent pregnancy is attacked with increasing ferocity. Consider the tragedy of the House of Tudor. King Henry VIII is perhaps the most famous case study in fertility anxiety. He tore England apart to get a son. For centuries, historians painted his wives as the problem. But modern bioarchaeologists have suggested that Henry's blood might have been the culprit. The reproductive history of his wives, particularly Catherine of Aragon and Anne Boleyn, fits the pattern of blood group incompatibility perfectly. Catherine of Aragon, a Spanish princess, and thus coming from that region of high R-negative Kel variants, was pregnant at least 6 times. Only one child survived infancy, Mary. The others were stillborn or died neonatally. Anne Boleyn followed the same tragic arc. A healthy first child, Elizabeth, followed by miscarriages of male fetuses at about 4 to 6 months. While we cannot draw blood from ghosts to confirm their Rh status, the clinical picture is that of a lethal womb scenario created by a genetic mismatch between the king and his queens. The bad blood wasn't a moral failing, it was a biological incompatibility. The devastation, was even more pronounced in the House of Stuart. Queen Anne of Great Britain, the last of her line, is perhaps the most tragic figure in the history of royal obstetrics. She was pregnant at least 17 times over 17 years, 17 hopes, 17 nurseries prepared. And yet, she died without a surviving heir. Only one of her children, Prince William, lived past infancy, and even he was sickly, dying at age 11. The rest were a litany of miscarriages and stillbirths. Contemporary descriptions of Queen Anne's stillborn infants are gruesome but telling. Physicians described babies born with dropsy, swollen with fluid, bloated, and pale. Today, we call this hydrops fetalis, and it is the most severe presentation of rhesus disease. It is almost certain that Queen Anne was Rh negative, or carried a similar antibody, and her husband, Prince George of Denmark, was positive. Her body, trying to protect itself, ruthlessly destroyed heir after heir. The result of this genetic war was the end of the Stuart dynasty and the rise of the Hanoverians. The course of British history was steered not by battles or parliaments, but by the microscopic antibodies in a queen's blood. Why was this so common in royalty? The answer lies in their marriage practices. European royals practiced aggressive consanguinity, inbreeding. They married cousins and nieces to keep titles and territories within the family. Geneticists call this pedigree collapse. In a normal, diverse population, recessive traits like Rh negative blood are scattered. But when you marry your cousin, you dramatically increase the chance that both of you carry the same recessive genes. However, the Rh dynamic adds a twisted layer to inbreeding. Inbreeding usually brings out recessive diseases, like hemophilia in the Romanovs. But Rh disease is caused by incompatibility. It requires a negative mother and a positive father. Royal intermarriage turned Europe into a genetic minefield. By constantly mixing these specific isolated gene pools, the Spanish Habsburgs, the British royals, the German houses, they were effectively concentrating the Rh negative gene and then pitting it against the Rh positive gene in the same bed. If a royal family had a high instance of Rh negative blood, like the Spanish lines, and they married into a line that was predominantly Rh positive, the statistical probability of a lethal match skyrocketed. They were creating the perfect laboratory conditions for hemolytic disease. They believed they were refining their blood, making it bluer and more noble. In reality, they were turning their wives' bodies into hostile environments for their own children. This perspective changes how we view the divine right of kings. It humanizes them in a tragic way. These monarchs believed they were cursed by God for their sins. They prayed, they went on pilgrimages, they consulted astrologers. Henry VIII believed his lack of sons was a biblical punishment for marrying his brother's widow. He couldn't possibly know that the problem was a protein on the surface of a red blood cell. The irony of the blue blood is palpable. The very trait that marked them as elite, the pale skin, the ancient Gothic Basque ancestry, carried with it the seeds of their own extinction. The purest blood turned out to be the most difficult to propagate. By the early 20th century, as the hemophilia gene, another blood curse, ravaged the Russian royal family and the Habsburg jaw deformed the Spanish kings, the era of royal inbreeding came to a crashing halt. But the damage had been done. The legacy of Rh negative blood in royalty is a testament to the power of invisible biology over human ambition. No amount of gold, armies, or decrees could stop the antibodies from crossing the placenta. It serves as a stark reminder that while we may build castles and invent titles, we are ultimately at the mercy of our own genetics. The blue bloods were not better or more divine. They were simply a specific genetic cluster, struggling to survive the same biological laws that govern the rest of us. And as the royal lines faded or branched out, the gene didn't disappear. It continued to mutate and drift. But to understand the true nature of this deletion, we have to stop looking at family trees and start looking at the code of life itself. We need to zoom in to the molecular level, to chromosome 1, to understand what exactly is missing in the Rh negative person. Because, as we are about to see, being negative isn't just about what you don't have, It's about a specific, violent break in the genetic chain. To truly understand what it means to be Rhe negative, we have to leave the macro world of kings, queens, and mountain tribes behind. We must shrink ourselves down to a size smaller than a single wavelength of light and enter the nucleus of the cell. We are going to look at the blueprint of the human body. We are hunting for a specific address in the library of our DNA, chromosome 1. This is the largest chromosome in the human genome, a massive archive containing over 3,000 genes and roughly 8% of all our genetic material. It is a bustling metropolis of data. But if you scroll down the long arm of chromosome 1 to a specific neighborhood known as 1p36.11, you will find the site of the accident. In the vast majority of human beings, about 85% of us, this location contains two functioning genes sitting right next to each other. They are called RHD and RHCE. They are like twin houses standing side by side. The RHD gene is responsible for producing the D antigen, that infamous protein we discussed earlier, the flag that triggers the immune system. The RHCE gene produces the other, quieter antigens, C, C, E, and E. Together, they form the rhesus blood group system. But in an Rh negative person, something dramatic has happened. If you look at that same address on chromosome 1, the RHD house is simply gone. It hasn't just been vandalized or boarded up, the entire structure has been ripped from the foundation. This is what geneticists call a complete deletion. In the genetics of European populations, and by extension the Basques and the Royals we just discussed, being Rh negative isn't a case of having a different gene. It is a case of having no gene. The 60,000 base pairs of DNA that make up the RHD instruction manual are missing. The strand of DNA simply skips over the gap where the gene should be and continues on to the next one. This is a profound distinction. Usually, when we talk about genetic traits, like blue eyes versus brown eyes, we are talking about alleles or variations. A blue-eyed person has a gene for eye color, but the code is slightly different than the brown-eyed person's. With Rh negative blood, it is binary. You either have the code or you have a void. This total deletion is what makes the Rh negative condition so biologically aggressive in terms of immune response. Because the Rhe negative body has absolutely no template for the D antigen, it has never seen it, doesn't have the blueprints for it, and cannot comprehend it. When it encounters that protein in a transfusion or a pregnancy, it views it as a totally alien structure. It reacts with maximum prejudice because the gap in its own DNA is absolute. So how did this happen? How did millions of people lose an entire gene? The answer lies in the architecture of the DNA itself. The RHD and RHCE genes are evolutionary siblings. Millions of years ago, there was likely only one Rh gene. At some point in our primate ancestry, the gene duplicated. It copied itself and pasted the copy right next to the original. This is a common way evolution creates new tools. It makes a spare copy that can then mutate and take on new jobs. So we ended up with two genes, RHD and RHCE, sitting head to tail on the chromosome. They are remarkably similar, about 97% identical in their sequence. But this similarity is a trap. When cells divide to create sperm and eggs, the chromosomes have to line up and exchange genetic material, a process called crossing over. It's like two zippers meshing together. Usually they line up perfectly. But because the RHD and RHCE genes look so much alike, the zipper sometimes gets confused. It misaligns. The machinery of the cell mistakes the RHD gene on one strand for the RHCE gene on the other. When the cell pulls the chromosomes apart, a mistake occurs. One chromosome ends up with extra copies of the gene. But the other chromosome, it gets the short end of the stick. It ends up with a deletion. The RHD gene is looped out and discarded. This event is what created the Rh negative haplotype. It is essentially a copy-paste error that happened thousands of years ago in the testicles or ovaries of an ancient ancestor. This specific deletion event, technically known as the rhesus box deletion, seems to be the single source for the vast majority of Rh negative people in Europe. It suggests that most white Rh-negative people are descended from a single individual or a small group of individuals in whom this error occurred. It didn't happen a million times randomly. It happened once or a few times and then spread like wildfire. But the plot thickens when we look outside of Europe. Remember how we said Rh-negative blood is rare in Africa and Asia? Well, when it does appear in African populations, the genetic mechanism is often completely different. In many black Rh-negative individuals, the RHD gene is actually still there. It hasn't been deleted. Instead, it has a mutation, a stop sign inserted into the middle of the code, a pseudogene, or a rearrangement that prevents the protein from being displayed on the cell surface. This means that Rh-negative is not one single thing. It is a phenotype, the result, but the genotype, the cause, varies by race and geography. The European deletion is just the most famous and widespread version. It paints a picture of humanity where different groups arrived at the same destination, no D-antigen, via different roads. The sheer scale of the European deletion brings us to a baffling biological question. What is the RHD gene actually for? In the human body, you rarely get to delete a massive chunk of code without consequences. If you delete a gene on chromosome 7, you get cystic fibrosis. If you delete a section of chromosome 4, you get Huntington's disease. Genes are usually vital. You pull one brick and the wall falls down. Yet, Rh-negative people are walking around with a 60th base pair hole in their genome, and they are fine. They are healthy. They run marathons, solve calculus problems, and live to be 100. The RHD gene appears to be, at first glance, completely disposable. We call this a null phenotype regarding the D antigen. The fact that 15% of Europeans lack this gene suggests that the D antigen is not essential for human life. It is an accessory, like a hood ornament on a car. The car drives fine without it. However, nature rarely keeps useless things, and it rarely tolerates the loss of useful things. The D antigen is a transmembrane protein. It weaves in and out of the cell membrane of the red blood cell. It is structurally complex. It looks like an ion channel, a gate that allows things to flow in and out of the cell. For decades, scientists assumed it was just a structural scaffold holding the cell together. But recent research suggests it might be doing something with ammonia transport or gas exchange. The fact that Rh-negative people lack this specific gate suggests their red blood cells must compensate in some way. They might be using a backup system. Or perhaps the RHCE gene works double time to cover the loss. This subtle difference in cell membrane architecture, the difference between a cell with the D gate and a cell without it, might be the key to understanding everything else. The strange disease resistances, the personality anecdotes, and the evolutionary persistence of the gene. The genetic deletion on chromosome 1 is the smoking gun of the Rhe mystery. It is a violent, definitive break in the chain of inheritance. To the fringe theorists, whom we will visit later, this clean cut looks suspicious, almost like genetic engineering. They ask, how can a gene just vanish? Why is the gap so precise? But to the geneticist, it is a beautiful, if chaotic, example of molecular evolution. It shows how a simple mechanical error in the cell's zipper can create a new category of human being. This void in the DNA, this missing paragraph in the instruction manual, is what defines the Rh-negative person. It is not an addition, it is a subtraction. And in the world of biology, sometimes what is missing is more powerful than what is present. By lacking this gene, the Rh-negative individual escapes the notice of certain viruses, but walks into the crosshairs of their own reproductive biology. It is a trade-off written in the nucleotides of chromosome 1. As we move away from the microscope, we have to ask, if this deletion is a loss, is there a gain somewhere else? If nature took away the D antigen, did it give something back in return? The answer seems to be yes. As we look at the modern world of virology and disease, it turns out that this missing gene might actually be a secret shield, guarding the Rh-negative population from some of the deadliest killers in human history. In the ruthless calculus of evolution, nature rarely allows a flaw to persist for tens of thousands of years, unless it is actually a hidden feature. We have spent much of this story looking at the heavy price of being Rh negative, the reproductive tragedy of the lethal womb, the incompatibility that turned a mother's blood against her own lineage. If this blood type were merely a genetic mistake, a broken zipper in the DNA, the brutal laws of natural selection should have scrubbed it from the human population eons ago. The fact that 15% of Europeans still carry this deletion suggests that there is a weight on the other side of the scale, a powerful survival advantage that compensates for the loss of infants. That advantage, it turns out, may be a distinct and formidable shield against the invisible armies of the viral world. To understand this, we have to look at how a virus operates. A virus is a biological burglar. It cannot reproduce on its own. It needs to break into a host cell and hijack the machinery to make copies of itself. But a virus cannot just walk through the wall. It needs a key. The surface of a human cell is locked, covered in proteins and receptors. A virus has evolved to recognize specific proteins, specific door handles, on the cell's surface. It latches onto them, turns the handle, and slips inside. This is where the missing D antigen becomes the hero of the story. Remember, the Rh-negative person is missing a massive chunk of protein on the surface of their red blood cells. To a transfusion doctor, this is a complication. But to a virus looking for a handhold, it might be a dead end. If a virus has evolved to grab onto the D antigen, or a structure closely linked to it, to gain entry or to travel through the bloodstream, the Rh-negative host presents a slippery surface. The door handle is simply not there. The burglar reaches out and grasps nothing but air. The most compelling evidence for this viral shield theory appeared in the early 21st century, centered around one of the most frightening pathogens of the modern era, HIV. In 2011, a landmark study was published in the journal Cell Host and Microbe that sent ripples through the scientific community. Researchers were looking at the viral load and progression rates of HIV patients across different populations. They noticed a startling correlation. Patients who were Rh negative were significantly less likely to be infected in the first place, and if they were infected, the virus progressed much more slowly than in their Rh positive counterparts. The study suggested that the presence of the Rh positive protein, the D antigen, might help the HIV virus stabilize itself or navigate the bloodstream more efficiently. Without that protein, the virus struggled to gain a foothold. It was like trying to climb a wall that had been polished smooth. This didn't mean Rh negative people were immune. They could still contract the virus, but their bodies put up a naturally stiffer resistance. Their blood was a less hospitable environment for the invader. This finding illuminated a massive piece of the evolutionary puzzle. It hinted that the Rh-negative mutation might have been conserved in the European gene pool because it acted as a survival mechanism against ancient pandemics. While we focus on HIV today, it is merely a modern proxy for the thousands of plagues that have swept through humanity over the last 50,000 years. It is highly probable that some ancient retrovirus, now long extinct, ravaged the human population in prehistory. This virus likely used the Rh protein as its key. The people who had the protein died. The people who had the mutation, the ones with the broken gene, survived. They lived to pass on their negative blood to the next generation. The lethal womb took some of their children, yes, but the plague would have taken everyone. This is the concept of heterozygote advantage, similar to how the sickle cell gene persists in Africa because it protects against malaria. The Rh-negative gene is a biological trade-off. You pay a tax in reproduction, but you buy insurance against infection. But the protection doesn't seem to be limited to just retroviruses. Other studies have hinted at similar resistances to other viral agents. There is data suggesting that Rh-negative individuals might have a different response to West Nile virus and potentially even certain strains of influenza. The mechanism here is slightly different and points to the personality of the Rh-negative immune system. We have established that the Rh-negative body is vigilant. It is quick to attack foreign blood in the womb. This suggests that the immune system of an Rh-negative person is trigger-happy. It is in a state of high alert. While this is bad for pregnancy, it is excellent for fighting off sudden infections. When a virus enters the body, the Rh-negative immune system often recognizes the non-self invader faster and launches a more aggressive scorched earth campaign to destroy it. However, this aggression comes with a cost. If your immune system is a hair-trigger militia, it shoots at everything, sometimes even things that aren't threats. This leads us to the other side of the health coin: autoimmune disorders. The same heightened immune response that protects Rh-negative people from viruses seems to make them more susceptible to allergies, autoimmune diseases, and inflammatory conditions. There is a statistically higher prevalence of allergies among the Rh-negative population. Their bodies react violently to pollen, dust, and dander, treating them like deadly pathogens. It is the curse of the super soldier immune system. It is so good at fighting that it starts wars in peacetime. There are also intriguing, though less definitive, links to other conditions. Some studies have suggested correlations between Rh-negative blood and higher rates of thyroid issues or certain autoimmune digestive disorders. It paints a picture of a physiological trade-off. The Rh-negative person is built for acute survival, surviving the plague, the infection, the sudden viral storm, but perhaps struggles more with the chronic internal friction of a hyperactive defense system. This viral resistance theory also helps explain the geography of the blood type. Recall that the Rh-negative gene is highest in Europe. Europe has historically been a disease incubator due to high population density, urbanization, and the domestication of livestock, which is where many viruses jump to humans. The Black Death, smallpox, the Spanish flu, these scourges hit Europe hard. It is plausible that the intense pressure of these repeated pandemics acted as a crucible, refining the population. If the Rh-negative gene offered even a 5% better chance of surviving a plague over thousands of years, that advantage would concrete the gene into the population, despite the reproductive downsides. Conversely, in populations that were more isolated or had different disease pressures, like the indigenous peoples of the Americas or Australia, the Rh-negative gene was not necessary, or the reproductive cost was too high to justify keeping it, so it vanished or never took hold. The broken gene on chromosome 1, therefore, is not a defect. It is a battle scar. It is a defensive modification. It's as if nature looked at the human body, saw that the Rh protein was a liability, a handle that viruses could grab, and decided to rip it off. The result was a blood type that was chemically quieter, harder for enemies to grapple with, but trickier to reproduce. This perspective shifts the narrative of the Rh-negative person from a medical victim to a genetic survivor. They are the descendants of the ones who walked out of the quarantine tents. When the pestilence came in the night, stripping the D antigen from the cell wall was like turning off the lights in a house during an air raid. It made the target harder to find. But the story of resistance isn't just about viruses, which are relatively simple genetic machines. The Rh-negative immune system also has a bizarre and complex relationship with a much smarter, more complex invader, a parasite that lives in the brains of billions of people, affecting their behavior, their reflexes, and perhaps even their culture. As we move from the viral world to the parasitic world, we find that the Rh-negative blood type might be the only thing standing between humanity and a microscopic puppet master that wants to rewire our minds. The viral shield is impressive, but the battle against the mind control parasite is where the science of Rh-negative blood begins to sound like science fiction. If the idea of a virus acting as a burglar is unsettling, the reality of Toxoplasma gondii is nothing short of a psychological horror story. We like to believe that our thoughts, our reflexes, and our personalities are our own, the sovereign territory of our consciousness. But nature has evolved organisms that view our brains not as a sanctuary, but as a cockpit. Toxoplasma gondii is a microscopic protozoan parasite, and it is arguably one of the most successful life forms on Earth. It infects roughly 1/3 of the human population. You might have it right now, sleeping in the neurons of your brain, and you wouldn't even know it. But if you are Rh negative, the way you interact with this intruder is fundamentally different from the rest of the species. To understand the Rh connection, we first have to understand what this parasite does. The primary host of toxoplasma is the house cat. The parasite can only reproduce sexually inside a feline gut. However, to get there, it often infects intermediate hosts, rats and mice. But a rat is naturally terrified of cats. The smell of cat urine usually sends a rodent into a panic. This is bad for the parasite, so toxoplasma hacks the rat's brain. It physically rewires the amygdala, the fear center. The infected rat not only loses its fear of cats, but becomes attracted to the smell of cat urine. It walks right up to the predator, offering itself as a meal. The cat eats the rat. The parasite completes its life cycle. It is a precision-guided suicide mission engineered by a single-celled organism. For decades, scientists thought that when humans were infected, usually by handling kitty litter or eating undercooked meat, it was a dead end for the parasite. They believed the parasite just formed harmless cysts in the brain and went dormant. But recently, a Czech evolutionary biologist named Jaroslav Flieger shattered that assumption. He spent years studying the subtle behavioral shifts in infected humans, and what he found was disturbing. Infected Rh-positive people started to change. Their reaction times slowed down. They became statistically more likely to be involved in traffic accidents. Their personalities shifted. Men became more aggressive and rule-breaking, while women became more outgoing and warm-hearted. It was as if the parasite was subtly tweaking the dials of their behavior, perhaps trying to make them more reckless, more likely to engage in risk, a ghostly echo of the suicide run it programs into the rat. But here's where the blood-type mystery reemerges. When Flieger and his team separated the data by Rh factor, the results were stunning. The Rh-positive subjects showed all the classic signs of the toxo effect: the slowed reflexes, the personality shifts, the deterioration of psychomotor performance. They were the puppets the parasite could pull the strings on. The Rh-negative subjects, however, were resistant. In fact, in some metrics, the Rh-negative individuals who were infected with toxoplasma actually performed better or showed absolutely no decline in reaction time compared to the uninfected control group. The parasite was there, sitting in their brains, but it couldn't touch the controls. The Rh-negative gene acted as a firewall. Why does this happen? The leading theory brings us back to the concept of the D antigen as a handle or a gateway. It is possible that the toxoplasma parasite utilizes transport proteins related to the Rh factor to move chemicals or signals into the host's neural cells. In an Rh-positive brain, the machinery is compatible. The parasite can plug in and start uploading its code. In an Rh-negative brain, the hardware is missing. The parasite shouts, but the neurons don't listen. Another possibility lies in that hypervigilant immune system we discussed. The Rh-negative immune system might keep the parasite in a much tighter chokehold. While it can't fully eradicate the cysts once they are in the brain, it might prevent them from releasing the dopamine-altering chemicals that change behavior. The negative blood creates a biological environment that is simply too hostile for the parasite to conduct its business as usual. This parasite paradox adds a fascinating layer to the survival of the Rh-negative gene. We previously asked why nature would keep a gene that causes reproductive death. The answer might be that the threat of manipulation was just as dangerous as the threat of childbirth. In a prehistoric world teeming with parasites and pathogens that targeted the brain, having a subpopulation that remained clear-headed, sharp, and immune to behavioral manipulation would have been a massive advantage for the tribe. Imagine a Paleolithic hunting party. They are all infected with a parasite that makes them slightly slower, slightly more reckless. They take unnecessary risks with a mammoth or a saber-toothed tiger and get killed. But the Rh-negative hunters, the ones immune to the mind fog, retain their lightning-fast reflexes. They survive. They bring the food home. They become the leaders. In this scenario, the Rh-negative gene is a clarity filter. It ensures that at least 15% of the population remains biologically sovereign, untouched by the chemical influence of the microscopic world. This resistance extends beyond just toxoplasma. It speaks to a broader theme of Rh-negative blood being a nonconformist biological state. Whether it is a virus trying to enter a cell or a parasite trying to rewire a synapse, the Rh-negative body often puts up a "do not enter" sign. It is less permeable to the biological noise of the environment. However, this resistance comes with a psychological intrigue. If Rh-positive people are more susceptible to the subtle nudges of environmental parasites, which affect billions of people, does that mean the human condition, as we know it, is partially shaped by these infections? And if Rh-negative people are immune to that influence, are they experiencing a slightly different version of reality? Are they the controls in a global experiment? The implications of the toxoplasma connection have fueled a quiet firestorm in the world of evolutionary psychology. It suggests that our blood type dictates not just our health, but potentially our temperament and our fate on the highway. It reinforces the idea that Rh-negative people are biologically distinct in a way that goes deeper than just transfusion compatibility. They are built differently, their brains are guarded differently. There is a darker flip side to this, of course. Some researchers have looked for correlations between Rh-negative blood and various psychiatric conditions, wondering if this immunity or altered wiring comes at a cost of mental stability in other areas. While the data is mixed and often controversial, the very fact that we are asking the question shows how deep the rabbit hole goes. We are no longer just talking about antibodies. We are talking about the architecture of the mind. This distinctness, this idea of being unhackable by earthly parasites, has naturally led the human imagination into wilder territories. If Rh-negative blood is so unique, so resistant to the things that afflict the rest of the primate family, and if it appeared so suddenly in the genetic record of Europe, could its origins be something else? For decades, while scientists were looking at petri dishes, another group of people was looking at the stars. They took the scientific facts, the lack of the monkey gene, the sudden appearance, the blue blood, the resistance to disease, and wove them into a modern mythology. They argue that if the Rh-negative gene doesn't fit the standard model of earthly evolution, perhaps it didn't come from Earth at all. The science of the parasite shield gave them the ammunition they needed. Rh-negative people are not just resistant to parasites, they are incompatible with the system. As we leave the microscope behind and step into the realm of cultural mythology, we have to address the elephant in the room, or rather, the astronaut in the room. The mystery of Rh-negative blood has spawned one of the most enduring and bizarre conspiracy theories of the internet age, the ancient astronaut theory. It is a narrative that takes the red mystery and turns it into a cosmic question. Are Rh-negative people the descendants of a hybrid experimentation? Is the missing link actually a star link? We have now arrived at the part of the story where the pavement of science ends and the wild jungle of modern mythology begins. Up until this point, we have relied on centrifuges, microscopes, and archaeological digs to trace the history of Rh-negative blood. We have spoken of mutations, deletions, and immune responses. But there is a massive subculture that looks at the exact same data, the lack of the monkey protein, the sudden appearance of the gene in Europe, the lethal rejection of offspring, and comes to a radically different conclusion. They argue that the reason Rh-negative blood behaves like an alien element in the human body is simple, because it is alien. This is the realm of the ancient astronaut theory, a narrative that has exploded in popularity over the last 50 years, largely fueled by the internet and the writings of authors like Zecharia Sitchin and Erich von Däniken. To the skeptics, this is pure science fiction. But to the millions of people who subscribe to these theories, Rh-negative blood is the smoking gun, the biological proof that humanity is not merely a product of terrestrial evolution, but a result of cosmic intervention. The core argument of the starseed or alien ancestry theory rests on a linguistic and logical leap regarding the name itself. When Landsteiner labeled the blood rhesus, He forever linked it to the primate. The logic of the fringe theorists goes like this. If the vast majority of mankind, 85%, has the rhesus monkey gene, they are the product of natural Earth evolution. They are of the Earth. But if 15% of the people are negative, if they lack the monkey gene, then they must not be related to the primate family tree. If they didn't come from the ape, they must have come from the stars. While any geneticist will tell you this is a misunderstanding of how taxonomy works, we share 98% of our DNA with chimps regardless of our blood type, the theory gains traction because it offers a dramatic explanation for the lethal womb. Why does a mother's body reject her own child? In nature, this kind of violent autoimmune rejection between mother and fetus is incredibly rare. You don't see it happening in dogs, cats, or horses to this degree. The ancient astronaut theorists argue that this incompatibility is evidence of a species barrier. They claim that Rh disease is exactly what you would expect if you tried to breed two different species together, like a horse and a donkey producing a mule. They suggest that Rh-negative people are a remnant of a separate hybrid lineage that is not fully compatible with the original Homo sapiens. To find the origin of this hybrid, these theorists turn to the oldest texts in human history, the Sumerian clay tablets. According to the translations proposed by Zechariah Sitchin, which are heavily contested by mainstream Assyriologists, the tablets tell the story of the Anunnaki, a race of extraterrestrials who came to Earth hundreds of thousands of years ago to mine gold. The Anunnaki needed workers, so they genetically engineered a slave race by splicing their own DNA with the DNA of the primitive hominids roaming the Earth. In this narrative, the Adamu, the first man, was a test tube baby. The theorists suggest that the Rhe-negative gene is the legacy of the Anunnaki blood. It is the divine spark, the piece of the creator code left in the genome. This, they claim, explains why Rh-negative blood is associated with the blue blood royalty. The kings claimed the right to rule because they were literally descendants of the gods from the sky. The exclusivity of the bloodline was an attempt to preserve this extraterrestrial connection. The theory also leans heavily on the Bible, specifically the mysterious passage in Genesis chapter 6 regarding the Nephilim. The text reads, "The sons of God saw the daughters of men that they were fair, and they took them wives of all which they chose. There were giants in the earth in those days, and also after that, when the sons of God came in unto the daughters of men, and they bare children to them, the same were mighty men which were of old, men of renown." For the fringe community, the sons of God, the Elohim, are the extraterrestrials or fallen angels. The daughters of men are the earthly humans. The offspring, the Nephilim, are the hybrids. The theory posits that the Rh-negative blood is the specific marker of the Nephilim bloodline. They argue that the great flood described in the Bible was actually a genetic purge, an attempt to wipe out these unauthorized hybrids and reset the human experiment. Noah, they argue, was chosen because he was perfect in his generations, meaning his genetic code was pure and uncorrupted by the alien mixture. This biblical sci-fi fusion helps explain the intense feeling of otherness reported by many Rh-negative people. In online forums and communities, thousands of Rh-negative individuals share anecdotes about feeling like they don't belong on this planet. They talk about having a low vibration tolerance, feeling overwhelmed by crowds, and having a deep, inexplicable longing for the stars. They report strange physical traits, lower body temperature, lower blood pressure, and extra rib vertebrae, a claim often repeated but rarely medically substantiated, and sensitive eyes. When Brad Steiger, a prominent paranormal researcher, termed these people star people, he gave a vocabulary to this sense of isolation. The blood type became an identity, a validation that their feeling of alienation was actually a biological memory of a different home. There is also a darker, more conspiratorial edge to the alien theory, the abduction phenomenon. Researchers into alien abduction phenomena have frequently noted a statistical anomaly. A disproportionately high number of people who claim to have been abducted by Greys or other entities report having Rh-negative blood. The lore suggests that the aliens are monitoring these specific bloodlines. Are they checking on their investment? Are they harvesting genetic material to repair their own degrading DNA? It turns out to be a self-reinforcing loop. If you believe you are special because of your blood, you are more likely to interpret a sleep paralysis episode as an alien encounter. But the statistical overlap remains a favorite talking point of the UFO community. Even the science of the Basque country is co-opted into this narrative. The theorists point to the Basque language, Euskara, which has no known relatives on Earth. They ask, if the language is an isolate and the blood is an anomaly, isn't it possible that the Basques are the original colony, the landing site? The Pyrenees Mountains, with their megalithic stones and ancient dolmens, are reimagined not as the refuge of hunter-gatherers, but as the stronghold of the sky gods. Critically, the ancient astronaut theory also attempts to explain the missing link in human evolution, the sudden leap in brain size and cultural complexity that occurred around 40,000 years ago. They argue that evolution moves too slowly to account for art, mathematics, and agriculture appearing so quickly. The Rh-negative mutation is viewed as the software update that facilitated this jump. It is the God gene. Of course, to a scientist, this is all smoke and mirrors. The sudden appearance of Rh-negative blood can be explained by the founder effect and genetic drift in isolated Ice Age populations. The rejection of the fetus is a quirk of immune system evolution, not a species barrier. The monkey name is just a label, and the Nephilim are theological allegories. But stories don't have to be true to be powerful. The alien blood narrative serves a psychological need. It takes a frightening medical reality, my body kills my babies, and reframes it as a secret superpower. It transforms a genetic deletion into a cosmic addition. It tells the Rh-negative person, you are not broken, you are advanced, you are not a medical anomaly, you are royalty from the stars. This mythology has created a modern tribe. In a world where people are desperate for identity and meaning, being Rh-negative has become a badge of mystery. It is a secret club where the membership fee is paid in antigens, and whether the origin is a mutation in a cave in France or a syringe in a spaceship over Sumer, the result is the same. A group of people who feel fundamentally different from the rest of the herd. However, we don't need aliens to find a mysterious connection between the Rh-negative people and the ancient world. We just need to look at the very real, very earthly migration of the tribes that carried this blood. As we leave the flying saucers behind and return to the soil, we find that the genetic thread connects the mists of Ireland to the sands of the Sahara. The Celtic and Berber link is not a myth. It is a historical reality that binds two of the most enigmatic cultures of the ancient world together, proving that the Atlantic Ocean was not a barrier, but a highway for the blood of the ancients. If you were to stand on the windswept cliffs of Moher in Ireland, looking out over the gray, churning Atlantic, and then transport yourself instantly to the sun-baked ochre peaks of the Atlas Mountains in Morocco, you would feel as though you had traveled between two different planets. One is a world of emerald green, mist, and relentless rain. The other is a land of arid stone, scorching heat, and desert winds. Culturally, they seem just as distant, the Celtic world of bagpipes and druids versus the Berber world of camels and minarets. Yet, if you were to take a syringe and draw blood from a shepherd in County Kerry and a nomad in the Rift Mountains, the microscope would reveal a stunning secret. They are, genetically speaking, long-lost brothers. This connection, often called the Celtic-Berber link, is one of the most fascinating revelations to emerge from the study of Rh-negative blood. It challenges our modern obsession with continental borders. We tend to think of the Mediterranean Sea as a dividing line between Europe and Africa, between white and black, between Christian and Muslim. But the blood tells us that for thousands of years, the sea was not a barrier. It was a bridge. It was a superhighway that connected a specific brotherhood of tribes who shared a unique genetic signature. including a remarkably high frequency of the Rh negative gene. To see this connection, we have to look at the blood map again. As we noted, the highest concentration of Rh negative blood is in the Basque country. But where are the runners-up? They are the insular Celts, the Irish, Scots, and Welsh, and the Amazigh, the Berbers of North Africa. In some Berber tribes, the rate of Rh negative blood is significantly higher than in the surrounding Arab or sub-Saharan populations, mirroring the frequencies found in Western Europe. This biological anomaly led anthropologists to dig deeper. When they looked at the Y-DNA, the paternal lineage, they found a shared marker, haplogroup R1b. This is the dominant male lineage in Western Europe, found in over 80% of Irish and Welsh men. But it is also found in significant pockets of North Africa, particularly among the Amazigh. This suggests a deep shared ancestry that predates the Roman Empire, predates the Arab conquests, and possibly even predates the written history of Europe. The story this genetic data tells is one of the Atlantic facade. It suggests that after the last ice age, as the glaciers retreated, the refuge population in northern Spain and southern France didn't just sit still. They exploded outward in two directions. One branch moved north, hopping along the coast in skin boats, populating Cornwall, Wales, Ireland, and Scotland. These became the ancestors of the Atlantic Celts. The other branch moved south, crossing the Strait of Gibraltar, which is only 9 miles wide, and populating the Maghreb, the northern coast of Africa. These became the ancestors of the Berbers. This explains why the physical anthropology of these two groups has often overlapped in confusing ways. For centuries, travelers to North Africa expressed surprise at finding Berber tribesmen in the Atlas Mountains with blue or green eyes, reddish hair, and pale skin that burned easily in the desert sun. These physical traits, which we typically associate with Northern Europe, are not the result of recent mixing with European colonists. They are indigenous traits, remnants of that shared Ice Age gene pool. Conversely, in Ireland and Britain, there is the persistent legend of the Black Irish, people with dark hair, dark eyes, and olive skin, yet who are fully Irish. Victorian historians, desperate to explain this non-Celtic look, invented the myth that these people were the descendants of shipwrecked sailors from the Spanish Armada in 1588. But modern genetics has debunked that story. There is virtually no Spanish DNA from the 16th century in the Irish gene pool. The Black Irish phenotype is ancient. It is likely the original look of the Atlantic hunter-gatherers, the same stock that gave rise to the Basques and the Berbers, before the lighter-haired, lighter-skinned farmers fully took over. The dark hair and the Rh negative blood are ancient partners. The connection goes deeper than just blood and hair. It echoes in culture and stone. Both the Atlantic Celts and the ancient North Africans were obsessed with stone. The coastlines of Ireland, Brittany, and Spain are dotted with megaliths, standing stones, dolmens, and stone circles. But if you travel to North Africa, specifically to places like Mamzora in Morocco, you find the exact same structures. The Mamzora stone circle, with its 167 monoliths arranged in an ellipse, looks like it could have been airlifted straight out of Salisbury Plain or County Cork. Archaeologists used to think these ideas developed independently, but the blood evidence suggests that the people who built them were in contact. They were part of a maritime culture that viewed the Atlantic not as a terrifying void, but as a pond. They traded, they traveled, and they intermarried. The R-negative gene was the passenger on these voyages, carried in the veins of the sailors and priestesses who navigated by the stars. This link also forces us to rethink the very definition of Celtic. In the 19th century, historians decided that Celtic meant the Hallstatt and Latène cultures of Central Europe, iron-wielding warriors who swept west. But geneticists now argue that the people of Ireland and Wales are not primarily descended from those Central European invaders. They are much older. They are the Atlantic Celts. Their language and culture might have shifted over time, but their blood is indigenous to the western edge of the world. They are the cousins of the Basques and the Berbers, not the Germans. There are even strange linguistic curiosities that tease at this connection. While Berber is an Afro-Asiatic language and Gaelic is Indo-European, linguists have noted peculiar similarities in syntax, sentence structure, between the two that don't exist in other European languages. For example, the way verbs and subjects are ordered in sentences in Irish and Welsh is different from English or French, but remarkably similar to Berber and ancient Egyptian. Some scholars propose a Hamito-Semitic substratum, the idea that the earliest languages of the British Isles were influenced by contact with North African seafarers. While controversial, the Rh negative connection gives this theory a biological backbone. The tragedy of history is that these two branches of the family tree forgot each other. As empires rose and fell, Rome, Carthage, the Caliphates, the British Empire, the Atlantic Brotherhood was severed. The people of the North became Europeans, and the people of the South became Africans. Religion and politics drew an iron curtain across the Mediterranean. The Berbers were Arabized and Islamized. The Celts were Anglicized and Christianized. The memory of their shared kinship faded into myth, but the blood didn't forget. In the maternity wards of Dublin and Casablanca, the same scene played out for centuries. Right, negative mothers losing their Rh-positive babies to the same silent ancient enemy. The gene remained a stubborn witness to their shared origin. It reminds us that the rigid racial categories of the modern world are recent inventions, often contradicted by the deeper, messier reality of human migration. Today, the Celtic-Berber link serves as a powerful symbol for Rh-negative people. It grounds their identity not in a single nation, but in a movement. It suggests that they are the descendants of a maritime aristocracy, a group of people who lived on the edge of the continents, facing the open ocean. They were the people of the stones, the people of the West. This distinct heritage brings us to a more subjective, yet endlessly fascinating aspect of the Rh mystery. We have talked about the science of the gene, the history of the migration, and the tragedy of the womb. But what about the people themselves? If you ask an Rh-negative person, do you feel different? The answer is often a resounding yes. Beyond the medical charts, there's a massive volume of anecdotal evidence and some intriguing scientific hints suggesting that this blood type comes with a specific set of physical and personality traits. Is there an Rh personality? Do they really have lower body temperatures? Are they more intuitive? Or is this just the Barnum effect where people see what they want to see? As we move from the expansive history of tribes to the intimate details of the individual, we have to navigate the blurry line between biological fact and the folklore of the phenotype. Because for many Rh-negative people, the blood isn't just a liquid. It's a lens through which they see the world and through which the world sees them. If you type Rh-negative traits into a search engine today, you will not be led to a medical journal, but rather to a sprawling, decentralized library of modern folklore. You will find yourself on forums, blogs, and social media groups where millions of people are comparing notes on their own bodies, building a composite image of what an Rh-negative person is supposed to look and act like. It is a laundry list of anomalies that reads less like a clinical diagnosis and more like the character sheet for a fantasy novel. The list is remarkably consistent. It claims that Rh-negative people possess piercing blue or green eyes, reddish or blonde hair, and a lower than average body temperature. It suggests they have low blood pressure, a slow pulse, and an extra vertebra or rib. It says they are highly sensitive to heat and sunlight, prone to inexplicable allergies, and, most intriguingly, that they cannot wear mechanical watches because their bodies emit a strange electrical charge that drains batteries and shorts out circuits. On the psychological side, the profile describes them as truth seekers, people with heightened intuition, intense empathy, a high IQ, and a pervasive lifelong sense of not belonging, as if they are merely visiting this planet rather than inhabiting it. So, how much of this is biological reality? And how much is the Barnum effect, the psychological phenomenon where people believe vague personality descriptions apply specifically to them? The answer, as is often the case with this blood type, lies in the gray zone between genetic coincidence and physiological fact. Let's start with the physical appearance. Is there an Rh negative look? Scientifically, the gene for Rh factor is inherited independently of the genes for eye and hair color. You can be Rh negative and have dark skin and brown eyes, just as you can be Rh positive and be a pale redhead. However, because the Rh negative gene is most concentrated in the Atlantic facade populations we just discussed, the Basques, the Celts, the Berbers, it is statistically much more likely to be found in people who also carry the genes for those physical traits. This is a phenomenon known as population stratification. If you are Rh negative, there's a high probability that your ancestors came from the misty coasts of Western Europe. Therefore, you are also likely to inherit the pale skin, light eyes, and reddish hair common to those tribes. The blood didn't cause the blue eyes, they are just traveling companions packed into the same ancestral suitcase. This explains why the piercing blue eyes trope is so persistent in the community. It is a correct observation of ancestry, even if it's not a direct function of the blood antigen itself. But what about the stranger physiological claims? The low body temperature and low blood pressure are harder to dismiss as mere demographics. While large-scale studies are scarce, anecdotal data from hematology nurses often supports the idea that Rh-negative donors tend to run cool. This could be linked to the metabolic differences we hinted at earlier. Remember, the Rh-negative person is missing a transmembrane protein that might be involved in gas transport or ion exchange. It is plausible that this absence forces the body to adopt a slightly different metabolic set point. If the red blood cells are operating on a different efficiency curve, perhaps the entire system runs at a lower idle speed, resulting in lower pressure and lower temperature. Then there is the extra rib. This is a persistent myth that likely stems from a misunderstanding of a real medical anomaly called a cervical rib, an extra bone arising from the seventh cervical vertebra. While it does occur in the general population, about 1 in 500 people, there is no concrete data linking it exclusively to Rh-negative blood. This specific claim likely belongs to the Nephilim mythology, an attempt to find a skeletal difference to match the genetic one. The most fascinating territory, however, is the mind. The Rh personality is described as hyperintuitive, analytical, and deeply resistant to authority. The community describes a scanning ability, the feeling that they can walk into a room and instantly read the emotional temperature, detecting lies or danger before a word is spoken. Skeptics call this confirmation bias. But if we look at the chromosomal neighborhood, there might be a genetic basis for some mental distinctiveness. This brings us to linkage disequilibrium. Genes that sit close together on a chromosome are often inherited together. They are neighbors who never move. The Rh gene sits on the short arm of chromosome 1, 1p36. This is a very busy neighborhood. It is one of the most gene-rich regions of the human genome, and it is a hotspot for genes related to neurological development and neurotransmitter function. Is it possible that the specific mutation that deleted the Rh gene also affected a neighboring gene involved in serotonin or dopamine processing? Or that the original Rhu negative haplotype included a specific variant of a brain gene that has been passed down alongside the blood type for 20,000 years? There was a study, albeit a controversial one, that looked at the correlation between blood types and personality using Cattell's 16 personality factors. It found some statistically significant spikes. Rh-negative men, for instance, tested higher on tough-mindedness and independence. Other smaller studies have hinted at higher IQ scores, though this is heavily influenced by socioeconomic factors. But perhaps the most compelling explanation for the Rh personality isn't genetic, but reactive. Consider the life experience of the Rh-negative woman historically. She is a woman whose body has betrayed her. She has likely suffered miscarriages or traumatic births. She has been told by doctors that she is incompatible with her husband. This kind of biological trauma can shape a personality. It can create a heightened sense of vigilance, a feeling of being other, and a need to question authority, specifically medical authority, to find answers. The seeker personality might be a survival adaptation to the medical mystery of her own life. Furthermore, we must return to the parasite shield. If the Rh-negative brain is indeed more resistant to Toxoplasma gondii and other neuromodulators, then the Rh personality might simply be the human personality, stripped of the parasitic static. If 85% of the world is seeing life through a filter of subtle viral or parasitic influence and 15% are not, then the 15% will naturally feel like outsiders. They will feel like they are operating on a different frequency because biologically they are. Their intuition might just be a clearer signal. And what of the electric body, the stopping of watches? This is the favorite anecdote of the Rh community. I can't wear a Fitbit. It dies in a week. Streetlights go out when I walk under them. Science struggles here. We are bioelectrical beings. Our hearts and brains run on voltage. Is it possible that the lack of the D antigen alters the electrical capacitance of the red blood cell membrane, changing the overall electromagnetic field of the body? It sounds like science fiction, but the cell membrane is a capacitor. Changing its protein structure changes its electrical properties. While there is no paper in Nature proving that Rh-negative people demagnetize credit cards, the sheer volume of reports suggests something is happening, even if it's just a higher level of static electricity generation due to skin chemistry. Ultimately, the phenotype of the Rh-negative person is a mosaic. Some of it is the Atlantic ancestry, the blue eyes. Some of it is the metabolic consequence of the deletion, the low temperature. Some of it is the psychological resistance to parasites, the clear-headedness. And some of it is surely the power of the tribe, the human need to find a pattern in the chaos. But whether these traits are hard-coded in the DNA or woven into the culture, they have created a distinct identity. To be Rh-negative today is to feel special. It is to feel like a diamond in the rough. And for many, this feeling of uniqueness comes with a heavy burden, the burden of being a golden donor. Because while the Rh-negative person may feel alien to the medical world, they are a commodity. And among them, there is a subgroup that is even rarer, a group so genetically unique that their blood is not just negative, it is null. This is the story of the rarest blood on earth, a liquid so precious and so dangerous that to possess it is to live in a glass cage. If being Rh-negative is like being a member of an exclusive club, then having golden blood is like being the only person in a room of 8 billion. We have spent this entire story discussing the absence of the D antigen, the single protein that separates the positive from the negative. But in the shadowy corners of hematology, there exists a blood type where the deletion didn't stop at D. It kept going. It wiped out the C, the E, and the E. It scrubbed the red blood cell clean of the entire rhesus system. This condition is technically known as Rh null, but in the medical world, it has earned a more mythical name, golden blood. To understand how rare this is, consider the statistics. There are millions of O-negative people. But since the discovery of Ra null in 1961, found in an Aboriginal Australian woman, fewer than 50 individuals on the entire planet have been identified with this blood type. It is so statistically improbable that university professors used to tell their students it was theoretically impossible. They believed that a fetus without the Rh complex simply couldn't survive gestation. But they do survive, and they walk among us carrying a substance in their veins that is both a miraculous gift and a terrifying curse. The nickname golden blood was coined by Dr. Thierry Perard, the director of the National Immunohematology Reference Laboratory in Paris, because to a doctor, this blood is more valuable than bullion. Here is the logic of its value. The Rh blood group system is huge and complex, consisting of 61 different antigens. Most of us have a mix of these. If a patient has a rare combination of antibodies against the Rh system, perhaps due to multiple transfusions or a complex pregnancy, finding a compatible donor is a nightmare. Rh null blood is the master key. Because it lacks all Rh antigens, it can be transfused into almost anyone with a rare Rh type without triggering an immune reaction. It is the universal solvent for rhesus incompatibility. If you have a patient dying of a reaction to a rare antigen like anti-little c or anti-big E, a bag of golden blood is the only thing that will save them. But for the person holding the gold, the situation is a trap. The Rh null individual is the ultimate giver, but they can receive from no one but themselves or another Rh null. If a person with golden blood is in a car accident in Tokyo and needs a transfusion, they cannot take O-negative blood. Even the universal donor blood of an O-negative person contains the other Rh antigens, like C or E, that the Rh null body has never seen. Their immune system would attack the O-negative blood just as violently as an Rh-negative mother attacks a positive baby. This creates a life of extreme fragility. People with golden blood live in a state of constant low-level vigilance. They drive carefully. They avoid extreme sports. They often wear medical alert bracelets that read like a death sentence to a paramedic. Rh null. No transfusion. Because of this danger, the few R null people in the world are part of a quiet, desperate network. There is an informal brotherhood, coordinated by the International Rare Blood Panel in Bristol, UK, and the lab in Paris. It operates like a biological Swiss bank. When an Rh null person is identified, they are encouraged to donate blood regularly, not for others, but for themselves. The blood is frozen and stored in cryopreservation vaults, kept at 80 degrees Celsius, waiting for the day the donor might need it. Occasionally, the call goes out. An Rh null person in Brazil might need surgery. The network activates. A unit of frozen blood is shipped from a vault in Amsterdam or Johannesburg. It is a logistical high-wire act. The blood has to clear customs, remain frozen, and arrive before the patient bleeds out. The anonymous donors who provide this blood are essentially keeping each other alive across oceans and borders. They are the only people on Earth who can save one another. But the burden of golden blood isn't just logistical, it is physiological. This condition finally answers the question we asked earlier. What is the Rh protein actually for? In Rh-negative people who are just missing the D antigen, the red blood cells look normal. They are the classic biconcave discs, like donuts without the hole punched all the way through. But in Rh null people who are missing the entire protein complex, the architecture of the cell collapses. Under a microscope, their red blood cells don't look like donuts. They look like stomatocytes. They have a mouth-like slit in the center. They are deflated. This deformity reveals the true function of the Rh complex. It acts as a structural scaffolding. It knits the cell membrane together, giving it the strength and flexibility to squeeze through the tiniest capillaries of the body. Without this scaffolding, the Rh null cells are fragile. Their walls are leaky. Sodium pumps in, potassium pumps out. The cells swell and burst easily. As a result, almost all Rh null people suffer from a chronic mild anemia. Their spleen is constantly working overtime to destroy the broken cells. They often live with a yellowish tint to their eyes, jaundice, and a fatigue that never quite goes away. It is a testament to the resilience of the human body that they survive at all. Nature has deleted a crucial load-bearing wall, yet the building still stands, albeit a little shaky. The existence of golden blood puts the ancient astronaut theories into a harsh, realistic perspective. While the internet loves to romanticize the negative blood as a superpower, the null condition shows us the raw, messy reality of mutation. It isn't a sleek upgrade. It's a deletion that leaves the body struggling to keep its shape. It is a glitch in the matrix, not a cheat code. Yet, there is a profound beauty in it. The story of R null is the story of human altruism at its absolute peak. Imagine being one of these 43 people. You know that your blood is precious. You know that somewhere in the world, a baby might be dying because they have a rare incompatibility that only your blood can fix. But you also know that every pint you give away is a pint you might need yourself if you trip and fall tomorrow. And yet, they donate. They fly to Paris to give their blood to strangers. They empty their own savings account of life to pay someone else's debt. This spirit of medical sacrifice is what ultimately turned the tide in the war against rhesus disease. While the Rh null donors are the special forces of the blood world, saving the rarest of the rare, a different kind of heroics was needed to save the millions of Rh-negative mothers and their babies. We have talked about the problem, the antibodies, the sensitization, the blue babies. Now we must talk about the cure. The discovery of the ROGAM shot is one of the greatest medical triumphs of the 20th century, a story that involves a dedicated doctor, a serendipitous experiment with prisoners, and the harvesting of a magic potion from the very blood that caused the problem. It is the moment where science finally outsmarted the lethal womb, turning a genetic tragedy into a manageable condition and ensuring that the red mystery would no longer be a death sentence for the unborn. By the late 1950s, the medical world had mapped the enemy. They knew the Rh factor existed, they knew how it killed, and they knew who was at risk. But knowledge is not a cure. The exchanged transfusions, draining and refilling the baby, were a desperate reactive measure. They saved lives, but they couldn't stop the disease from happening. The holy grail was prevention. The doctors needed a way to stop the Rh-negative mother's immune system from ever waking up in the first place. The breakthrough came from an unlikely trio in New York City: Dr. John Gorman, an Irish-born pathologist, Dr. Vincent Frieda, an obstetrician, and Dr. William Pollock, a British immunologist. Their idea was so counterintuitive, so backwards, that when they first proposed it, their colleagues laughed at them. They proposed giving the mother the very thing that killed the baby: antibodies. The concept was based on a quirky rule of the immune system called antibody-mediated immune suppression. Think of the immune system like a security guard patrolling a building. If the guard sees an intruder, the baby's Rh-positive cells, he radios for backup, and the whole police force, the mother's permanent antibodies, arrives to smash the place up. But Gorman and Frieda wondered, what if we sent in a fake squad first? What if we injected the mother with a dose of pre-made Rh antibodies immediately after birth? The theory was that these injected antibodies, the passive ones, would quickly hunt down and cover up the baby's Rh-positive cells that had leaked into the mother's blood. They would arrest the intruders and clear them out before the mother's own security guard even noticed they were there. Once the job was done, the injected antibodies would naturally fade away. The mother's immune system would never sound the alarm, never make its own permanent antibodies, and the next baby would be safe. It was a stealth mission. To prove this worked, they needed human subjects. In a move that would be ethically complex today but was common in the 1960s, they turned to the inmates of Sing Sing Correctional Facility. They needed Rh-negative men who had no risk of pregnancy, to act as the testing ground. The prisoners volunteered, knowing the research might save future children. The experiment was grueling. The doctors deliberately injected the prisoners with Rh-positive blood to see if they could trigger an immune response. Then, they treated one group with their experimental gamma-globulin shot, the early version of ROGAM, and left the other group alone. The tension in the lab was palpable for months. When the results came back, they were flawless. The untreated prisoners had developed the deadly antibodies. The treated prisoners. Their blood was clean. The shot had acted as a perfect shield. But to make this vaccine on a global scale, they needed a raw ingredient. They couldn't synthesize the antibodies in a test tube. They had to harvest them from human blood. They needed a donor who had incredibly high levels of anti-Rh antibodies flowing through their veins. Enter James Harrison. If the Sing Sing prisoners were the test subjects, James Harrison was the factory. An Australian man, Harrison had undergone a major chest surgery as a teenager in 1951, requiring massive blood transfusions. Those transfusions saved his life, but they also accidentally sensitized his blood. His body was overflowing with the specific Rh antibody needed to make the vaccine. When researchers realized what was in his veins, they asked him to donate. Harrison didn't just agree, he dedicated his life to it. Because his blood contained the magic ingredient, he became the primary source for the anti-D immunoglobulin in Australia and beyond. For over 60 years, James Harrison sat in a chair every week or two, letting doctors harvest his plasma. His antibodies were processed, concentrated, and turned into millions of doses of the vaccine. It is estimated that James Harrison, the man with the golden arm, saved the lives of over 2.4 million babies with his donations. His single biological quirk, combined with the brilliance of the New York team, turned the tide. In 1968, the drug was approved in the United States under the name RhoGAM. The impact was instantaneous and miraculous. The incidence of Rh disease plummeted. The wards of weeping women, the sections of hospitals dedicated to mothers losing their babies, began to empty out. The terror of the second pregnancy evaporated. All it took was a single shot given to the Rh-negative mother within 72 hours of giving birth. The development of RhoGAM is arguably the most successful prophylactic program in the history of medicine. It turned a genetic curse into a manageable checklist item. Today, an Rh-negative woman gets a shot at 28 weeks and another after delivery, and she rarely has to think about it again. The lethal womb was disarmed not by changing the woman's genes, but by outsmarting her immune system. However, the success of RhoGAM has had an interesting side effect. It has made us forget. Because the babies stopped dying, the mystery of the Rh factor faded from the public consciousness. We stopped asking why the incompatibility existed because we had fixed the consequence. The red mystery became a footnote in obstetrics textbooks, but the question remains. We have masked the symptoms, but the underlying biological reality is still there. 15% of the population still walks around with a deletion on chromosome 1. They still carry the ancient blood of the Basques and the Celts, and nature, in her infinite wisdom, is still maintaining this genetic divide. As we reach the end of our story, we have to pull back the camera one last time. We have looked at the how, the when, and the who. We have traversed from the monkeys to the aliens, from the mountains to the microscope. Now we must face the final lingering question. Why? In a universe that ruthlessly edits out weakness, why are the Rh-negative people still here? Is this blood type a remnant of a bygone era, slowly fading away, or is it a banked fire, waiting for a future where its unique properties might be the very thing that saves us all? We have traveled a long way together, tracing the path of a single drop of blood through the corridors of history. We started in the candlelit rooms of the 19th century, weeping over empty cradles, and we journeyed through the high-tech labs of New York, the misty peaks of the Pyrenees, the royal courts of Europe, and even into the speculative realms of ancient astronauts. We have seen how a microscopic deletion on chromosome 1, a simple missing paragraph in our genetic instruction manual, has the power to shape dynasties, dictate medical destiny, and spark global conspiracy theories. But as we close this book, we must return to the fundamental question that hangs over the entire story like a persistent fog. Why? In the grand, ruthless casino of evolution, the house always wins. Nature is an unsentimental accountant. If a trait costs too much, if it kills too many offspring, if it makes reproduction too difficult, it is usually liquidated. It is scrubbed from the ledger. By all conventional logic, Rh-negative blood should have been one of these failed experiments. The lethal womb was a massive tax on survival. For thousands of years, Rh-negative women paid a heartbreaking price, losing child after child to a war they didn't know they were fighting. And yet, the gene is still here. It didn't just survive. In some parts of the world, like the Basque Country in Ireland, it thrived. It stood its ground against the incoming waves of Rh-positive farmers. It persisted through famines, wars, and migrations. This persistence is the ultimate proof that the Rh-negative gene is not a mistake. It is a strategy. The answer to the why is likely found in the concept of balancing selection. Nature loves diversity, not for the sake of beauty, but for the sake of security. Imagine the human species as a massive computer system. If every single computer runs the exact same antivirus software and a hacker finds a way around it, the entire network crashes. Every screen goes black. But if 15% of the computers are running a different operating system, maybe an older one, maybe a weirder one that lacks the specific port the hacker is trying to access, then the network survives. The 15% keep the lights on. The Rh-negative people are that backup system. By deleting the D antigen, nature created a biological minority that was chemically distinct. When the great plagues of the past swept through, the retroviruses, the protozoans, the unknown pathogens of the Ice Age, they likely keyed into the standard Rh-positive protein structure. The positives were the easy targets. The negatives, with their missing proteins and their trigger-happy immune systems, were the firewalls. They were the ones who walked out of the cave when the fever broke. The tragedy of the incompatible pregnancy was simply the cost of doing business. Nature was willing to sacrifice a certain number of babies in mixed marriages to ensure that a reservoir of resistant humans remained in the gene pool. It was a cold, calculated trade-off, individual heartbreak for species survival. This perspective transforms the Rh-negative person from a medical anomaly into a guardian. They are the designated survivors. They carry the ancient blood not because it is obsolete, but because it is a proven shield. The Pyrenees paradox isn't a paradox at all. It's a bunker. The Basques and the Celts preserved this genetic line in their isolated corners of Europe, holding onto it like a biological seed bank, just in case the world needed it again. And in a way, we did. When the rhesus disease crisis hit the modern world, the solution, the RhoGAM shot, didn't come from a synthetic drug. It came from the blood itself. It came from the antibodies of people like James Harrison. The poison became the cure. The very reactivity that made the blood dangerous in the womb was harnessed to save millions of lives. The Rh-negative system proved that its aggressive nature could be turned into a tool for salvation. As we look to the future, the red mystery takes on a new relevance. We live in an era of emerging diseases, of zoonotic viruses jumping from animals to humans, of antibiotic resistance. We are realizing that genetic homogeneity is a weakness. We need our outliers. We need the people whose bodies react differently, whose cells are built with different scaffolding, whose immune systems shoot first and ask questions later. The Rh-negative gene, with its strange resistance to toxoplasma and HIV, might hold secrets we have yet to fully unlock, secrets that could lead to the next generation of vaccines or therapies. But beyond the science, there is the human story. The saga of Rh-negative blood is a testament to the complexity of identity. We are not all the same under the skin. We are a mosaic of ancient migrations, forgotten mutations, and invisible adaptations. The blood that flows through our veins is a liquid history book. For the Rh-negative person, that history is written in a script of resilience and difference. Whether you believe they are the descendants of the Ice Age hunters, the lost tribes of Israel, or the star travelers of the Nephilim myth, the feeling is the same, a sense of being set apart. And perhaps that feeling is necessary. The world needs its sensitives, its seekers, its people who feel the static electricity of the universe a little more intensely. The folklore of the Rh personality, the intuition, the independence, the feeling of being an outsider, might just be the cultural echo of a biological truth. If you are built to survive on the edge, you will likely see the world from a different angle. So the next time you look at a blood test result and see that little negative sign, don't see it as a lack. Don't see it as a deletion. See it as a badge of honor from an ancient lineage. See it as a trace of the original Europeans who weathered the glaciers. See it as the mark of the survivor. The mystery of the rhesus factor has been solved in the lab, but its poetry remains. It reminds us that we are still evolving, still adapting, and still carrying the ghosts of our ancestors in every heartbeat. The red mystery is no longer a source of fear, but a source of wonder. A reminder that in the vast, complex story of human life, even the things that seem like errors are often the most important parts of the design. The candles in the 19th century room have burned down. The crying in the nursery has stopped, replaced by the quiet breathing of a healthy child. The fear has been replaced by understanding. The blood flows on, carrying its secrets into the future, silent and red and vital. And as the story ends, we can finally rest, knowing that the mystery is not a curse, but a part of the miracle of being alive.